our last day before the exam. We have a final exam on Wednesday at 9 o'clock, not 9:30. Wednesday is at 9 o'clock in this room, as far as I know. They haven't changed that. So, first thing is, do you have any questions? It is comprehensive. It's going to go all the way back to chapter 1 through what we've done. And it will include chapters 11 and 12 that have not been tested before. Okay? Yes, ma'am.
I have a question about, do we not come Tuesday then?
You do not come on Tuesday. Tuesday there's 2 days of final exams. So Tuesday and Wednesday are final exams. And you do not come on Tuesday. That's probably the 8 to 9:15 class. They made the final exam schedule real simple. And I sent you all a copy of it. Okay. Other questions? Probably should have questions, yes.
Can you explain a little bit further the anatomical and the responses?
In terms of,
Just like differences between
in terms of natural selection? Okay. Let me see if I can figure out what's making the noise on the phone. I turned off. Anatomical would be having a longer neck. Anatomical would be having longer legs. Anatomical would be, it might include coloration. Coloration would kind of depends both on anatomy and physiology, so that's a tough one to plug into a specific category. It's anything that has to do with a structure. It might be how a joint is put together, something like that. It could be maybe the physical size of the brain. Physiological would be things like how much insulin does an organism make, or how does it respond when sugar is consumed? Does it produce the appropriate amount of insulin? That would be a physiological. How do the muscle fibers react to acetylcholine, which is the neurotransmitter that causes muscle contraction? So things like that would be physiological. Behavioral would be things like, does the squirrel run up a tree? That would be behavioral. Are there mating rituals that you have to go through? Taking care of young would be behavioral. So things like that would be behavioral. Does that help clarify? Okay.
Can you go over the disruptive and stabilizing selection and all that?
Okay, the three categories are they're really outcomes of selection. The three outcomes are directional, which means things shift if you have a normal bell-shaped curve. And what do you know if you have a kind of a bell-shaped curve? What do you know about the genetics of that particular quality?
Isn't it more like evenly distributed? It's more evenly distributed.
Okay, but what does that tell us about the genetics? Is it controlled by one gene or by multiple genes? By multiple genes. Okay, almost always. If you get like skin color, there's kind of a range of skin color, for example, in a lot of organisms. Sometimes there's a range of heights. Sometimes there's a range of physiological response. Anything like that is probably controlled by multiple genes. Okay? So if you have a particular character, and the example that was given was like fur color in rabbits or rats or something. So if you have a change in the environment that instead of favoring the one that's in the middle, starts favoring one of the extremes, the whole curve kind of shifts one direction or the other. And I think the example I gave is with global climate change, there's not as much snow in southern Canada as there used to be. So rabbits in southern Canada have begun shifting from white fur to more brownish fur because there's less of the season that there's snow, so it's less advantageous to have white fur. So that would be directional. Disruptive is like what we saw on the Galapagos, where you have usually two distinct extremes, and you lose the preference for the middle. On the mainland, there were lots and lots and lots of food sources. So having a medium-sized beak was great because you could eat some of the seeds and some of the flowers and some of other stuff. But once you got on an island that was relatively new in terms of geographic time, or geologic time, I guess, better term, then no longer was that true. There was only maybe one or two food sources. So the middle is no longer favored and the extremes become favored because either they're going to eat seeds and need a strong short beak, or they're going to eat nectar and they need a long skinny beak. But a medium-sized beak is not good for either one of those food sources. So you lose the middle. So that's called disruptive, or there was another term for it, selection. And then the third kind was the stabilizing selection, where the extremes are not good and the middle becomes better. And human birth weight is one of those, where it's a fair, again, without medical intervention, having a birth weight between about 6 and 8 pounds is generally the best. Okay, somebody has a hand up.
I was just asking for disruptive. Does that one lead to like speciation?
Say that again.
Does disruptive lead to speciation?
It is more likely to lead to speciation than the other two, because they're so extremely different. So over a period of time, they're less likely to continue to interbreed. So disruptive is most likely to lead to speciation. Not in the short term. It's going to take a long, long time. But eventually. Other questions? Let's, go ahead.
Can you go over bottleneck and founders effect?
The what, the founders?
The founders and bottleneck?
The difference between the founders and the bottleneck, in the bottleneck you have a small, both of them are examples of what? What larger category? Genetic drift. They're both examples of genetic drift. And genetic drift is a type of what?
Mutation. Or like,
What's the larger term? There's four types of it. Four types of microevolution, four mechanisms of microevolution. Genetic drift is one of them, and two of the subcategories under genetic drift are bottleneck and founders. Genetic drift requires a small population, not a large. You're not going to have a large population. You've got to have a relatively small population, and it has to be a chance event. So with a bottleneck, you've got your population, and something happens, usually a natural disaster of some sort happens, that eliminates the vast majority of the population. And you're only left with a tiny fraction of what you started with. And that tiny fraction of what you started with is not necessarily representative of the whole population. And since what's the definition of microevolution? How do we know when microevolution has happened? when there is what?
The change in like a shorter amount of time.
A change in what, though? How do we actually measure it? I'm hearing words, but I can't tell who's saying them. A change in what? What we measured with our worms. Change in phenotype, but that's not what defines microevolution. What? Allele frequency. We know we have microevolution when there's a change in allele frequency. What is an allele? These are questions that we ask on the senior comps and people don't get them sometimes. That's why I'm trying to get to. What is an allele? It's a genetic form. It's basically a different form of a gene. You have different alleles for the same character. And so it's those different alleles, it's a DNA sequence that makes up a gene. And if it's different than another DNA sequence that makes up for the same character, then we call those two alleles. It's the little b and the big B. Okay? So that's an allele. So where were we? The original question, bottlenecking. Okay, so if your final population that's going to repopulate has a different allele frequency than your original population, then we say microevolution has taken place, and the mechanism was genetic drift, and the specific was bottleneck. A founder effect is when you have individuals from a population that leave that population and go somewhere else and start a whole new colony. Now that's different than gene flow, which is when individuals from a population leave and join another population. In founder, a group of one population leaves and starts a whole new colony somewhere else. That's what happened with the potatoes. They took potatoes from, I don't know exactly where, somewhere in the Western Hemisphere, and took them to Ireland. It was a good climate, it was an introduced species, but it was only a few alleles compared to potatoes on the whole continents of North and South and Central America. So they didn't have all that allele frequency. So they had a much smaller gene pool. Even though they made lots of potatoes, they had lots and lots and lots of potatoes. It was a much smaller gene pool. And that's a big problem with endangered species. There's not enough genetic diversity left in the natural population so that those organisms will survive an environmental event that could wipe out whatever that was they were sensitive to. Okay, good questions. Did I answer your question? Other questions? Chapter 1, this is the way I would suggest you study. Go to each chapter. What was the main idea? Think about, remember I think I told you the other day, think horizontally. Think about what were the big ideas in chapter 1.
Can we go over one just so I get an idea of how to do the other chapters?
Yeah, we're going to. Chapter 1's not a great example.
Okay.
But then go down a couple layers. But don't go all the way down. Don't do vertical where you go all the way down into one topic and miss everything else. Okay? So let's take chapter 3. Chapter 3 was on basically nutrition, largely. It was on the macromolecules, right? What are some of the big ideas in chapter 3?
The 4 different types of the large molecules.
Right, there's 4 different types of macromolecules, but before we get to that, were there any big ideas that were consistent through all 4 of them?
They all have hydrocarbons.
Not really. They all have carbon, hydrogen, oxygen. How do we put them together? How do we put them together? Think about the word. How do we put them together? We take monomers and make polymers. How do we do that? Dehydration synthesis. That's a big idea in chapter 3. Dehydration synthesis. How can you remember which one is putting it together and which one's breaking it apart? What's it called when we break them apart? Hydrolysis. Look at the words. Think about what you're saying. How can you tell which one's which?
In hydrolysis, the lyse means to break.
Lyse means to break, so hydrolysis is breaking something. What does the word synthesis mean? Putting stuff together. Okay? So dehydration synthesis is putting stuff together by doing what? Not adding, dehydration. Removing water. Okay? What does hydrolysis mean? Breaking things apart by using water. Okay? So think about the terms, and that will really help you a lot. Okay? That's why I keep going through breaking down words for you, because I think if you'll put them together that way, it'll help. Okay? So that was a big idea. You should know dehydration synthesis and hydrolysis. You should know that all the macromolecules are made by putting together smaller molecules. Now, 3 of the 4 categories make polymers. 1 does not make a polymer. Which category does not make polymers? I'm sorry?
Nucleic acids.
Nope. Nucleic acids are long chains of nucleotides. One of them doesn't make, they're big molecules, but they don't keep adding monomers together.
A lipid?
What?
A lipid?
A lipid. A lipid still does dehydration synthesis because it has to put together a glycerol with 3 fatty acids, for example, in a triglyceride. But it doesn't then hook all those things together to make a big fat. You still have separate. The macromolecules, they're made of smaller molecules, but they're not polymers in the sense that you keep adding units to it. And they're broken down with hydrolysis. Okay, so what are the 4 categories?
Carbohydrates.
Carbohydrates,
Proteins.
proteins, lipids, and nucleic acids. What 3 elements do all of those have in common? Carbon, hydrogen, oxygen. You should know that. Carbon, hydrogen, oxygen. Almost any molecule associated with living systems, almost all are going to have carbon, hydrogen, oxygen. Okay? What else do proteins have? Nitrogen and sulfur. How can you remember the sulfur? What happens when you burn hair? It stinks. Okay? Almost everything that stinks has sulfur in it. Okay? Sulfur stink. Okay? Put those together. The reason natural gas smells is because they put sulfur-containing compounds called mercaptans in it. So you'll know you have a gas leak. Okay? All right. What 2 additional elements do nucleic acids have? Nitrogen and phosphorus. So both proteins and nucleic acids have nitrogen. Proteins have sulfur, nucleic acids have phosphorus. How can you remember nucleic acids have phosphorus?
If you know the way nucleotides are put together.
If you know that ATP, think about it, ATP is a molecule you should know some about, right? What does ATP stand for? Adenosine triphosphate, okay? ATP is a nucleotide. It has phosphate. That's where we get our phosphate. Okay? Then, under each of those, you should know what the monomers are. It probably wouldn't hurt to know the calories. I probably wouldn't ask you directly, but I might ask you which one has the most calories. I expect you to know the foods, okay? There is no reason that anybody in this room doesn't know what food has carbohydrates, proteins, and fats in it. You learned that in kindergarten or before, okay? So, that's an easy question. You know when you're eating what has what. You go to the cafeteria, do they still have little signs up? They probably don't. They used, last year they had little signs, for several years they had signs up to tell you how much.
They have it in the app,
They have them in the app? Okay.
I see they're on there now.
They do have some? Okay.
Shows the calories.
Shows the calories.
Like, I think something like, I remember either like a dairy or something with sign.
Yeah. Yeah. Okay. Can you think of anything else you should know about the,
Their monomer,
I'm sorry?
people's monomer, the lipid people.
Yeah, you have to know the monomers. Okay? So that would be, so learn the big picture stuff, then go down a couple levels. You don't have to go into as much detail as I expected for each individual exam.
So would we have to know like unsaturated, saturated fat?
You should probably have a vague sense. I probably wouldn't ask you to identify one or something.
Do you know about how many questions the final will be?
Generally, it's an in-class one, so it'll probably be something around between 90 and 100 multiple choice questions. There will be one short answer that everybody has to answer. And then there'll be probably, typically 4 other short answer questions that each have a choice. And I usually, just to make life easy on me, those are optional. If you choose to do the short answer questions, you leave off 12 of the multiple choice questions of your choosing.
Wait, we said it again. I'm sorry.
Okay. Basically, you can either do all the multiple choice or all the short answer. Those are your choices. For each short answer, there's going to be 2 options. Describe cellular respiration, describe photosynthesis. Pick one. Okay? That might be an example. Okay? Of the choices. So either you do all 4, well, all 5, because the first one everybody has to do. The other 4 you can choose to do or not do. But if you choose to do them, then you have to select 12 multiple choice questions to not answer. And those questions, you'll just take your pencil and mark through B, C, and D on those 12 questions. If you do all the multiple choice and you do all the short answer, I will not grade the short answer. So you can't do both. I mean, you can do both, but I'm not going to count them. I only give you that option because I know some people are just way better at the short answer than they are at the multiple choice. But to be able to leave off multiple choice questions, I expect good answers on a short answer. You're going to have to put down more than a sentence. Okay? If you expect to get anything. And basically, each one then is worth 3 of the multiple choice questions. But you can't do 2 short answer and leave off 6 multiple choice. It doesn't work that way. All or none. Not all or none. All of one or the other. Yes.
Did you say we get to pick the short answer, or everyone has to?
Each one has it's not like there's 8 questions and you can pick 4 of them. There's a choice for number 1, there's a choice for number 2, there's a choice for number 3, and a choice for number 4. So you only have to do 4 of them, 4 additional, plus the one everybody has to do. But I expect a regular, a good answer. So basically, I would say, unless you can do a good job on 3 of the short answers, 3 out of 4 of the short answers, I would just do multiple choice. Yes?
Would the short answer be like examples that we have in other examples?
Yeah, they would be similar to questions you had on other exams. I'm not necessarily saying they have to be complete sentences. I'm not teaching grammar, okay? But they have to be readable and understandable, address the question, and I have to be able to read your writing, okay? If you're gonna do the short answer questions, realize that I am 63 years old, and my vision is not what it was when I was your age. So if you write really, really tiny or really, really badly, I can't read it. I've been known to hand it to my husband and say, can you read this? And he's bad, because he can't even read his own handwriting.
Is there a way that we can open up like previous exams so that see what we got wrong?
If you want to come in, bring your computer and come in, I will open up your specific exam for a few minutes to let you look at it. But I'm not going to open them up broadly.
What about like the on paper?
You can come and look at those.
What's the best time to come look at them?
Tomorrow, I probably won't get here early. I would say maybe 11 tomorrow. Between 11 and 12 probably tomorrow would be good, or after maybe 3:30. Or this afternoon, pretty much I usually go to lunch. Right after this class is fine. I usually get back from lunch about 1:30, 1:45, so anytime after that, and I'm usually here till 5:30. So you might want to shoot me an email. But I'll probably take you, if there's not a class, take you across the hall and let you look at them in there. But you cannot take pictures, you cannot copy down questions. But you can look at them and take notes. Use your master in biology. Use those, what are they called? The dynamic study model. Yes.
I use, I don't know if you have this in your book, it's like a QR code scanner. And then like if you scan it, it takes you to a little video on like this particular part of the chapter. I thought it was really good.
Oh, okay. It was on the master in biology?
No,
or
it was like in your book.
in the physical
Yeah.
textbook? Yeah. Hadn't even noticed those. I've used QR codes more in the past 6 months than I have ever, because you go to some place and you have to scan the QR code to get a menu or get something. Take something back. Take your Amazon return back to Kohl's, and I think they will give QR code to scan.
So for the short answer, the one that will be like required, will it be just like the other short answers, or will it be like more complex, like multiple,
It's not something you can study for.
Okay.
But the short answer questions will each be worth 10 points. So I expect a good solid answer.